symbiosis deviates from the more orthodox Darwinian view of evolution, primarily by the mutation and selection of individual species. Nevertheless, everyone now agrees that Margulis was right some of the time. In particular, her theory of endosymbiosis, by which a bigger cell swallowed a smaller one 2 billion years ago to create the nucleus, is compelling and almost universally accepted today. And the idea has been extended because modern plants, animals, and fungi are made of cells with many kinds of internal structures that appear to have been earlier generations of small cells. So, for example, the mitochondria that act like tiny power plants, and the chloroplasts that harness the sun's energy in photosynthetic organisms, those tiny structures in eukaryotic cells, along with the cell nucleus, all hold their own DNA. When a cell divides, each of those structures also divides. Furthermore, these and other organelles in complex cells have their own cell membranes. In 1968, when Margulis first proposed that each of these organelles evolved from earlier, simpler cells that were engulfed and ultimately co-opted to perform specific biochemical tasks, many biologists, well, they thought she was nuts. But when the mitochondria were found to possess their own DNA, the pendulum swung in her favor, and that is now the conventional wisdom. Based on the fossil record, our best guess is that the transition to modern eukaryotes began about 2 billion years ago and set the stage for much more complex, multicellular life like animals and plants to arise. By the Neoproterozoic era, complex eukaryotic cells with nuclei were well established. They had experimented for 1 billion years with microbial mats and other multicellular colonies, and they were about to begin a new era in cooperative biology. More than 600 million years ago, single-celled organisms learned how to grow and move as a single entity. In short, they learned to become animals. The earliest unambiguous fossil evidence for an ecosystem that was dominated by animals, as opposed to microbes and algae, comes from the so-called Ediacaran period. That's a time interval that began about 635 million years ago, which was shortly after the second of the 3 great Neoproterozoic snowball Earth events. The first of these rather distinctive symmetrical fossils to be recognized came from a terrain of 580 million-year-old rocks from the Ediacaran region in southern Australia. That's where the Ediacaran fauna got its name. These fossils represent an array of soft-bodied animals, some of which are easy to imagine as distant relatives of jellyfish and worms. Fossil impressions are not easy to interpret, but they do have pleasing shapes, some like ornately lined pancakes called Dickinsonia, or fancifully striated leaves like Charnia. Unlike almost all microbes, these are big, obvious fossils. Some of them are as much as 2 feet across. Closely related fossils that have been found in many places around the world in rocks from a 65 million year span between about 610 and 545 million years old. By far the most remarkable Ediacaran site is probably the 633 million year old phosphate-rich Doshantou Formation of southern China. This amazing deposit holds fossilized clumps of microscopic cells that are interpreted as animal eggs and embryos. Individual clusters look just like the early stages of animal development today, with 8 or 16 or 32 or even 64 cells arranged in a little ball. These structures, which grew in shallow seas just after the second of the big Neoproterozoic global glaciations, appeared identical in every respect to modern animal embryos. In this way, the severe snowball hothouse climate cycle ultimately played a central role in the evolution of the living world as we know it today. In fact, it would be accurate to claim that the modern biosphere owes its very existence to that interval starting 800 million years ago when Earth reached a climatic tipping point. And the co-evolution of the geosphere and the biosphere has only increased in complexity and consequences in the time since. What's perhaps surprising is how very long it took for life to venture onto land. New chemical evidence obtained from shallow water sediments deposited about 575 million years ago suggests that the great algal blooms of that post-glacial time period may have begun the transition. Those are the earliest rocks that show evidence for green photosynthetic algae that had the ability to survive on swampy land. That suggests that for the first time in Earth's history, there were patches of green, slimy algae above ocean level along some shorelines. With these new stable populations of photosynthetic life, atmospheric oxygen levels would have continued to rise along with an increasingly protective stratospheric layer of ozone. That radiation barrier effectively shielded Earth's solid surface from harmful ultraviolet solar radiation, and that protection was an essential prelude to the emergence of the terrestrial biosphere of plants and animals. Nevertheless, even after the establishment of that ozone layer, it took perhaps another 100 million years for life to crawl onto land. Indeed, for many millions of years, the most significant biological innovations took place in the shallow waters of sunlit coastal seas. Based on the Ediacaran fauna, jellyfish and worms and microbially precipitated reefs appear to have dominated the post-glacial oceans for perhaps 40 million years. The many varieties of soft-bodied animals appear to have fed on seafloor detritus in what seems to have been a kind of ecological status quo, maybe for tens of millions of years. That period of stability didn't last, though. It was dramatically altered by about 530 million years ago, when numerous groups of animals learned how to construct their own protective shells out of hard minerals. It's quite a mystery how this evolutionary development came to be. Of course, microbial life had been depositing mineral layers for billions of years in the form of reef-like stromatolites. But stromatolites are more like a simple layered platform in which the microbes could grow and attach. That's a real advantage in a rough tidal zone, but stromatolites aren't shells. Animal shells are different because they provide a protective armoring with an elaborate mineral architecture. So we don't know how, but we do know when. Following the Gaskir's glaciation, the last of the Neoproteroid ice ages, that's about 580 million years ago, an unknown animal evolved the ability to grow its own protective hard parts out of commonplace minerals, most often calcium carbonate or silica. Calcium was available in essentially unlimited quantities in the ocean, so marine life had an advantage here over freshwater life. And you can imagine how this innovation provided a tremendous advantage in the struggle for survival. Predators aren't going to waste the energy to break a tough shell when they can eat a nice soft-bodied worm. The realm of ocean life was transformed as the rule quickly became make your own shell or die. Consequences for the fossil record are absolutely astounding. The resulting layers of sediments are for the first time packed with amazingly diverse life forms: corals, brachiopods, bryozoa, mollusks, and my favorites, the trilobites. The sudden transition from soft-bodied to shelly fauna constitutes such a dramatic change in sediments that the time has been called the Cambrian explosion. I have to confess that Cambrian explosion is quite a misleading term, at least in the sense that the transformation to a biosphere with shells was not at all sudden. The timing is more like that of the mineral explosion we discussed several lectures ago. In fact, the Cambrian explosion is literally another stage within mineral evolution as well, with a new propagation of minerals such as calcite, aragonite, and dolomite, which are all carbonate minerals, as well as the phosphate hydroxyl apatite and opal. And it took many millions of years of what might be thought of as experimentation in biomineralization to catch on. As long ago as 580 million years ago, there were sponges that had hardened spines made of hydrated silica or calcium carbonate. Some of those animals are preserved in the fossil-rich Doshanto Formation in southern China. That's the same place where those fossil embryos were discovered. And by about 550 million years ago, which is the tail end of the Ediacaran period, the fossil record holds a variety of hardened tube-shaped structures formed from carbonate minerals. Those armored burrows have been interpreted as the protected homes of worm-like creatures in the ocean floor. The oldest clearly recognizable ecosystem